Herzlich willkommen zu einem weiteren Gespräch aus meiner Lab Leak Interviewserie. Beim ersten Mal habe ich mit dem deutschen Lab Leak Forscher Dr. Valentin Brüttel gesprochen und heute spreche ich mit einem amerikanischen Forscher, nämlich Jim Haslam, der letztens auch bei Peter McCullough war. Bis vor kurzem galt Jims Theorie als etwas exotisch, auch unter den Lab Leakern, denn Jim gehört zu der Sorte von Lab Leakern, die den Virusursprung anders als die meisten nicht in China, sondern in den USA vermuten. Und er kann dies auch sehr gut belegen. Darüber hinaus sagt Jim, bei SARS-CoV-2 handelt es sich um einen selbst ausbreitenden Fledermaus-Impfstoff. Und das klingt zugegebenermaßen erstmal ziemlich abgefahren. Bevor jetzt die ersten auf Durchzug schalten und sagen, glaub ich eh nicht, würde ich bitten, sich wirklich mit den Fakten von Jim einmal auseinanderzusetzen, denn ich bin der Meinung, an diesen Fakten kommt man eigentlich nicht mehr vorbei, wenn man das Thema wirklich verstehen und durchdringen möchte. Das Interview hat schon im April stattgefunden und ich komme erst jetzt dazu, es zu veröffentlichen. Es ist auch leider nochmal Zoom und technisch nicht ganz perfekt. Zum Beispiel sitzt Jim zu nah an der Kamera. Aber ich habe mich trotzdem entschieden, genau dieses Gespräch zu veröffentlichen, denn es lässt sich in der Form auch einfach nicht nochmal reproduzieren. Warum es so lange gedauert hat, hat auch einfach damit zu tun, dass ich eine halbe Doku draus gemacht habe, weil ich sehr viel Bildmaterial reingeschnitten habe. Jim hat auch ein Buch geschrieben, und zwar dieses Buch hier. Ich habe es noch nicht gelesen, aber dafür seinen gesamten Substack-Blog, jeden einzelnen Artikel. Und kann es wirklich sehr empfehlen, wenn ihr mit Jims Artikeln einsteigen wollt, empfehle ich diesen hier. Ich habe eine ganze Nacht durchgelesen und war extrem fasziniert. Das Gespräch heute wird in 2 Teilen erscheinen. Teil 1 ist heute, Teil 2 dann im neuen Jahr. Und es gibt jeweils 3 Versionen davon, damit es bei diesem Thema wirklich nicht an der Sprachbarriere scheitert. Auf Englisch mit deutschen Untertiteln und einmal komplett deutsch synchronisiert, wo ich Andreas Sparberg sehr herzlich für die Synchronisierung danke. Jetzt erstmal wünsche ich euch sehr viel Spaß bei diesem Gespräch und ich hoffe, ihr seid von diesem Thema ähnlich fasziniert wie ich. Greetings, welcome to my interview. Today is with me Jim Haslam from United States. He is a lab leak researcher. And the title of your newsletter is Reverse Engineering the Origins of SARS-CoV-2, a consensus sequence of events from Wuhan to DC to Chapel Hill to Hamilton. And a disclaimer, this is a series of posts not so much to discuss lab leak, but what leaked. Yeah, what leaked?
What leaked? It was a vaccine that leaked. That is my overall arching theme. The virus was an animal vaccine. It was not a human vaccine. It was not a human virus. It was not designed for us. It was designed for an animal, a distant mammalian species, a relative of ours that has nothing to do with us. It was a complete accident because of that. But the virus was a vaccine is essentially everything I've been trying to write towards for the past couple of years. And I think that once we admit that what the virus is, we can have a little more mature conversation about where it came from and who designed it, where we can avoid this mishap from ever happening again.
Yeah, great. Thank you. I know you for quite some time, since 2021, but I've kind of lost track of all these events because I was into other topics. And then when I, in December 2023, discovered your newsletter again, I thought like, wow, there's a whole lot of things going on in the US, and in Germany, we don't even have a clue what happens. So this interview is a little bit about bringing the American lab leak discourse to Germany. First of all, I have to ask you, like, why should we even talk about the lab leak in 2024? Because I tell you honestly, in Germany, it's not a big issue. Sometimes we get some Twitter posts from the United States. Okay, there's a select committee on the coronavirus pandemic, and there has been a Barrack hearing and a DESAC hearing, but there is not a media discourse about this in Germany. Why should we still talk about lab leak in 2024?
It's a great question. Everyone's going to have their own answer. I think it's relevant because none of us want to relive that ever again. So, and a lot of people don't want to discuss it because they think they'll never understand who did it or why they did it. And I'm basically here to tell you I know exactly who did it and I know exactly why they did it. I can give you a name, I can give you a time, I can give you a place. So that's kind of why I don't think people want to talk about it. It's this nebulous Chinese black hole that they don't want to dive into, or this nebulous biodefense American boondoggle that they do not want to see into. And I don't want to talk about, nation states. I don't want to talk about organizations. I want to talk about 6 individuals. 6 people blew up the world. That is going to be, the overarching theme of all this. We can count them on basically one hand if we really want to get down to it. There's 5 people that did this to us. So it is just 5. Each one of them is individually innocent, but collectively they created COVID. And it's very easy to follow the story if you narrow down your list of suspects to 5. It's not even my theory. It's what they wrote. It's what they said they were going to do in 2018. We just cross-reference what they said in 2018 versus the results of what leaked from the lab in 2020, and it aligns perfectly. It is one of the most funny, easy jigsaw puzzles to solve. It was an R&D accident. It's very easy for 5 people to keep something quiet, because even if the 5 people came forward and said we did it, they kind of did, no one would believe them. So my story is their story. That's kind of what I want to talk about. I think it's still relevant because I don't think any one of us want to live through that ever again. The lockdowns, the vaccines, the mandates, it was just a nightmare. And it will happen again if we do not talk about the 5 people that did this to us.
I think that it's important to talk about the lab leak issue because the entire pandemic preparedness industry, instead of being defunded after what happened, have even collected more grants and go on their nice conferences again. They're all on the stage again. It's rock and roll, it has not ended, and they could even collect more taxpayers' money. So I think it's going totally in the wrong direction also to connect climate change narrative to zoonosis narrative, because they will always sell us the next lab leak as the next zoonosis and feed all the other agendas with this narrative. Because your publication is called a consensus sequence, I first wanted a little bit to talk about the controversy in the lab leak scene, because obviously there is not so much consensus. Like, there's such a thing as a mainstream lab leak narrative, and then there is a counter thesis, kind of. The mainstream is, okay, there is such a thing as a lab leak, but it came from China. And the counter thesis is kind of, yes, a lab leak, but origin United States. And you kind of belong to the latter group, right?
Yes. I separate lab leak, which I consider to be Wuhan, versus lab origin, which I consider to be US labs. You have to understand that it's an international industry. They do not care what your passport is. They do not care your background. They only care about where you are and what you can deliver. And, a small group of people get together internationally, which is what virology is. It is an international industry, truly the most international of all industries I've ever come across in my life. I've been in a couple of industries that are international, but this one is by far the most. So people share, knowledge and resources and samples and vaccines all the time, because they're basically on different parts of the globe trying out different antidotes for different lab species. So we need to separate lab leak, what I consider to be what most people believe, the epicenter of Wuhan. And we need to separate lab origin, because if a virus is engineered, wherever it spilled over into the human population does not tell us where it was engineered. So that's a bit difficult to grasp at first. But yes, that is essentially what the Substack is all about.
And I find it funny, as you described it, they send virus on filter paper. I mean, it's not funny. To be honest, that should not happen. But it's totally unfamiliar with people who are not in the virologist world, how these people act, that they kind of travel with viruses over borders, and that kind of borders don't exist for them.
Yes, they do not even think about a border. They do not even care about it. They smuggle samples and send each other, genomes all the time through the mail on filter paper. I did not understand until I got into this debate. But the Christian Drostens of the world share samples with the Shi Zhenglis of the world, and vice versa. It's a truly international industry, and they're laughing at us for not figuring out who did it. You can almost see them laughing at us. Look at these idiots, these peasants that couldn't even figure out who did this. So as you said, they're on the stage still, collecting the tax dollars under there's some golden light. They are laughing at us. That's why I think people should get a little worked up over. It's one of the greatest stories of our lifetime. The people on stage, the experts that the media called on, were the people that created this thing.
Tell us a little bit about DARPA DEFUSE. What is DARPA DEFUSE, and how is it connected to DESEC, EcoHealth Alliance, BARIC, and Fauci?
Okay, so from 2020 to 2021, everyone had a theory, and everyone was just kind of throwing, spitballs against a wall and seeing what would stick. I had my own theory. I thought it was the Chinese military. I thought it was, some sort of biodefense vaccine for SARS for the troops in a trench. That was my theory. The beginnings of the lab leak debate, I really do believe, start on September 21st, 2021, because none of us knew anything. We had nothing to work on. We really didn't. There was no gotcha. There was no evidence. There really wasn't, if we're being honest. But on September 21st, 2021, a U.S. Marine walked off the DARPA campus. His name is Major Murphy, and he leaked the DARPA DEFUSE grant. It was the loser's bid in a DARPA project. DARPA is this it's this cutting edge, leading edge brain of the Pentagon, the Department of Defense, the U.S. Department of Defense, the $1 trillion a year American empire. As a small group, inside of that little group is the big brains, and inside the big brains was this big idea that we're going to vaccinate animals, in China. And one of the ideas came from Peter Daszak. Daszak, as it's pronounced. Father's Ukrainian, and he grew up in Britain, had an interesting lifestyle there, from what we can gather. And he moved to New York City in 2000, and he got on top of this small little think tank called Eco Health Alliance, which most of us have all heard of now. And he grew it. He grew it from one or two projects to probably about 10 or 20 projects, but expensive projects, big projects. USAID Predict, NIAID with Tony Fauci, and the DoD. He was bidding on this DARPA project, and it was worth $14 million. This is big dollars for anyone that wants to talk about this. And inside this bid was a pre-confession to the SARS-2 crime. Basically, one University of North Carolina professor saying, I, in 2018, will insert fear and cleavage sites into live coronaviruses and test them on live bats in Wuhan. So this was a pre-confession to the crime. Essentially, inside this bid that Peter Daszak has submitted in 2018, they are predicting exactly what will emerge in Wuhan in 2020, down to the percentage point. They were off by 0.1%. They said 20% different. It was 20.1% different. Fear and cleavage site, check. Less than 5% different than RATG13, check. So I consider the DARPA DEFUSE document, even though it was not funded by DARPA because it was expensive, it was a pre-confession to the SARS-2 crime. And we can now narrow down our list of suspects from 8 billion people down to 5. So we can start there and work our way out. But then things are a little less head spinning. They're a little less, confusing. And now we can at least put names to suspects.
I think in the US, it's kind of the dominant theory. So it's not that fringe theory, and probably we will never find out. But I think it's a consensus in the United States that there was a lab leak.
Yes, 75% of us believe lab leak, but 100% of those 75% think the Chinese did it. It gets very uncomfortable when I start talking about the Americans that did it. It's not easy to understand something that emerged in Wuhan had American fingerprints. It's not pleasant, oh, we all knew China blew up the world. No, America did it, no, not America. There's very few Americans involved in this. Peter Dajak's from Britain. One of the other guys is from the Netherlands. There is one American, North Carolina. That's really about it. There's another one. The other one's Australian. The other one's Chinese. So it's truly an international team. We all did it. We got to get away from the collective idea that, China or America or all that. It really doesn't have much to do with anything. No CIA, no, MI6, no one knew anything. That's the funny thing about this. No one knew anything until DARPA DEFUSE leaked on September of 2021. So that's why I treat the document.
So this is one consensus point. This is at the heart of that issue. And this is why kind of the lab leak became the dominant theory, at least in the US. Then there is also a consensus about the issue of cover-up, that there has been a cover-up. Can you tell us a little bit about the Slack messages that leaked, and also the Fauci emails, and what do these Slack messages tell us?
So the Slack messages were a private group of world-class evolutionary virologists in January and February of 2020 discussing the overwhelming evidence for a Wuhan lab leak. And they can speak bluntly because they think it's private. This is January, February of 2020. Evolutionary virologists have gotten together, 4 of them, and they have basically come to the conclusion that this thing is engineered. And the only reason they know it's engineered is because what I call a whistleblower inside the Wuhan Institute of Virology has published a natural genome, a natural sequence, very close to SARS-2, but not quite the same. And it exposed the very particular part of SARS-2 that was engineered, called a fear and cleavage site. So there is a woman inside the Wuhan Institute of Virology named Shi Zhengli. She's actually everyone's suspect number 1. She's published the genome. If you look closely at what they're talking about in the Slack messages of Christian Andersen he's Danish, but he lives in California. There's Eddie Holmes. He's British, but he lives in Australia. There's Bob Gary. He's American. Then there's Andrew Rambot, I believe, and he's British. So it's truly an international team having these February 1st, 2020 conversations about how the genome's engineered. And it's so obvious. It's ridiculous. It's not even really up for debate. And what do we do about this? They want to call the FBI. The group really wants to call the FBI. And so the group got together. They sent Anthony Fauci an email saying, we find the genome inconsistent with expectations of evolutionary theory. And they're like, we want to call the FBI. We've now learned, Anthony Fauci of America did not call the FBI. He called Ralph Baric. So Ralph Baric on the phone. So I don't bring it too far along, but in January, February, every evolutionary virologist knew that the genome was engineered. Those same evolutionary virologists published a paper showing that it was not engineered.
The cover-up is kind of a consensus in the lab leaker scene. Then also the furin cleavage site. You talked about this right now shortly. And what does furin cleavage site mean? Like, what does it do in the body? Why does it make SARS-CoV-2 so special? And why were the virologists freaking out about it so much when it was published?
To quote one of the virologists, it is the magic sauce of SARS-CoV-2. Whether the virus is natural or unnatural, it's why it's circulating in humans. Is this tiny little thing called a furin cleavage site. Is essentially, it's a scissor that cuts the genome in half as it exits one cell so it can re-enter another one. So it allows it to daisy chain. The virus can now move through various cells of your body and organs of your body and work its way through every single one of them. And when it's done with you, it moves on to the next body. It's essentially what makes this thing move. It is the horsepower. It is the engine. It is a very beautiful piece of engineering. It's almost like the key fob on a Mercedes. It is just perfectly engineered and designed to sit right in the middle of this SARS-2 genome. And it was very hard to see it at first until the Wuhan Institute brought attention to it. So this is where it gets a little counterintuitive, but they almost like put their cards on the table and said, look, compare the two genomes, my genome RATG13 versus your genome SARS-CoV-2. And right in the middle was this thing called the furin cleavage site, almost as if they put, the designer put his hands in the genome, pulled it apart, put the furin cleavage site right in the middle. It is what they were all looking at when the Wuhan Institute published this genome called RATG13, which is very close, like 96% close to SARS-2, but not exactly. At that time, that's when Kristen Drosten and a bunch of other virologists looked at the genome very closely and they're like, that is suspicious because this type of virus does not have a furin cleavage site. There is no, it's called a group 2B coronavirus, a sarbecovirus. They do not have a furin cleavage site. So this was the anomaly, and this was the main point of discussion during the debate with Kristen Drosten, Eddie Holmes, Christian Anderson. Why did they even publish RATG13 was something else the group is talking about. Why do they want us to virology hand over the murder weapon and show us the evidence? Like they're very confused. But Christian Drosten entertained the idea of a furin cleavage site because he even talked about, the restriction site flanking the furin cleavage site. So he goes into detail. He's suspicious, but he's playing the company line. You don't want to rock the boat. It's a virus in China, and no one even knows what Wuhan is at the time. It's February 1st, 2020. So it's going to burn out, even if it did leak from the lab. And it got worse and worse and worse, and it got so big that you really can't do much, besides just stay quiet and call it a conspiracy.
You also mentioned right now the other issue that most lab leakers, I would say, agree upon, which is the restriction enzymes, which also point towards a lab origin. And you said the only restriction site that really matters is BSAX1. Why is it so?
It's the one Christian Drosten was talking about. I like Christian Drosten as far as an arbiter of least lab leak. And he answers some of the, he hasn't answered my questions. I haven't interacted with him, but he's answered a couple of lab leaker questions. He said the Wuhan Institute of Virology don't have a genetic engineering system that could create something like SARS-2. It's just they do not. And one of those details he said is they have what they call virocells in the Wuhan Institute of Virology, and virocells delete the furin cleavage site. Another one is that they can only copy the existing genome. They cannot create a new genome. SARS-2 is a new genome. They can only copy an existing genome. If the Wuhan Institute of Virology creates an engineered genome, it has more than 6 segments. It has 7, 8, 9, because they're so bad. They have to chop up the genome even more. Essentially, you start off with an RNA virus, you copy it into DNA, you rearrange it, and then you copy it back into RNA, and you recreate the virus as a live genome that way. So when you're doing that little conversion back and forth, you chop up the 30,000 nucleotides into smaller pieces to work with it easier. And the Wuhan Institute of Virology has to use 7, 8, and 9 pieces, but the SARS-2 genome has 6 pieces. Those 6 pieces are cut up by 5 restriction sites that Valentin Bruttel has published. He did change the lab leak debate. He proved that essentially this thing has a eerie, spooky spacing in it. Those 5 restriction sites create 6 somewhat evenly spaced segments. The Wuhan Institute of Virology uses 7, 8, or 9. The University of North Carolina, Ralph Barrick, uses 6. There's a 7th restriction site inside the SARS-2 genome called a BSAX1, and it flanks the furin cleavage site that we keep talking about. Drosten apparently noticed it early on because he's talked about it in the public press. If they're playing with a genome, they leave a signature, they leave a trace. Sometimes they can hide it, but they usually do not hide it. It's an accident. It leaked was an accident. We need to admit that. And it was a prototype, and it left the fingerprints in it so they could rearrange it in the future to do what they wanted it to do, to infect lab animals, not us, to infect lab animals. So you leave your fingerprints in it so you can go back and find them, change them, rearrange them. And in the process of doing that, I've heard it called Legos. Most of the discussion on February 1st with Christian Drosten was about restriction enzyme sites and the furin cleavage site. And around the furin cleavage site was a BSAX1 perfectly on either side of this furin cleavage site, which is not supposed to be in this virus, as if someone left it in there so they could change it out with another one in the future if they needed to. So it screams engineering. It's a Western, a clue though. And it was a very fascinating point for researchers in the West. 0 papers in China showing interest in a furin cleavage site in before 2020. There's not one. There's one talking about a chicken coronavirus in Beijing, and there's another one like a protein in Shanghai. But there's not, there's 0 interest in Chinese literature in a furin cleavage site in a 30,000 nucleotide coronavirus. There's just not one. I mean, it was studied in the West, Western biolabs. It was a point of fascination. But there's just not one paper to be found in all of China. 1.5 billion people, and none of them showed interest in it. It's just, they're 20 years behind the West when it comes to biotechnology, maybe 30 or 40 years. I too thought, it leaked. I too thought it had something to do with the Chinese. I thought it was Chinese junk, and I thought it would burn out, and it never did or never will, simply because of that furin cleavage site. So this was the smoking gun that someone's been playing with this thing, and someone that knows what they're doing has been playing with this thing, because you could not even tell it was engineered at first. It was impossible. The genome was published on January 10th of 2020, but no one suspected engineering until January 24th of 2020. When what I call the whistleblower, I know it's hard for people to hear that, Shi Zhengli inside the Wuhan Institute of Virology, put her cards on the table and said, this is what I got, do what you want with it. She actually brought attention to her lab. Her lab is the BSL-2 in downtown Wuhan. If you read anything about lab leak, you read about her lab. You read about Shi Zhengli. She's the bat lady. She's this tiny, petite Asian woman who basically is standing in the big, giant BSL-4 spacesuit, even though she doesn't really spend much time in that. She works in a relatively low-level lab doing low-level research. She's a batshit collector on the US payroll collecting samples. That was her job description.
And her patents are about how to keep bats alive in the lab conditions. And the patents of Ralph Barrick are about the furin cleavage site, right?
Yeah, it shows what they're interested in. You patent what you're interested in. You patent what you're thinking about. You can get a glimpse into someone's mind if you look at their patents. And Shi Zhengli's patenting how to feed live bats in the lab. And she goes into great detail, a 12-hour light cycle, 2 worms a day, humidity, temperature, 15 degrees Celsius, like 60% humidity. It has to replicate a bat cave inside of a lab. So she keeps live bats alive. That's why she's special. That's why Wuhan's, the only reason Wuhan's a story is because of live bats. These are very special bats that she can keep. No one else can. Other labs have tried to do it. America tried, failed. Australia tried, failed. Singapore tried to keep these Chinese bats alive, failed. They're very picky eaters. They only eat worms. They do not eat fruit. They're very high maintenance. They're like Chinese pandas. They don't feed and breed in captivity very well. You just can't do it, unless if there's a panda shipped to America, it dies, because it just doesn't work. But the panda can be kept alive in a Chinese zoo. That's what the Wuhan Institute of Virology was able to do. They could keep live bats alive. But if you read Western literature, they're infatuated with the live bat. Christian Drosten's infatuated with the Chinese horseshoe bat because it was seen as the source of SARS-1. There's a lab in Germany. It's called FLI that keeps live bats alive in a BSL-4. It provided some pretty stellar evidence for a lab leak, not just a lab leak, a Western creation of a lab leak. So, live bats are kept in German labs. It's not abnormal. That's what this whole thing was about. Live creatures, inside of a lab. It is a point of infatuation, obsession. It is all they think about is where do these viruses come from? But that's where the industry was in 2018, 2019. We need to study live bats. We need to study where these viruses come from, not just SARS, not just MERS. Remember the Middle Eastern camel virus? There was a Nipah. It's a big outbreak in Singapore in 2000. There's something called Hydra in Australia. All these lethal, Ebola is the big one. That one comes from bats. What's the other one? Marburg. All these lethal viruses that people are scared of and they try to, run away from, they come from bats. And the researchers were studying live bats. And that's what Shi Zhengli had, live bats inside the lab. It's the only reason why I believe Wuhan is a story.
And why bats? I read something interesting that it's 20% of the mammal species. That's why they consider it the reservoir population. And it's never infecting humans directly, but there's always an intermediary. And like in the SARS-1 epidemic, it was Lin Wang who found out a bat is the origin in the animal kingdom.
Yes, they are the reservoir host for all these nasty pathogens. There's 1,300 species of bats out of 5,000 mammals. So they are 1/4 of every mammal on the planet is a bat. So
Wow.
they are a very distant relative of us humans. This is where it gets kind of
Mhm.
icky. But birds are dinosaurs. They have nothing to do with us. Bats are mammals. They have fur. They have neocortex. They have glands. They have they milk. They only have 1 or 2 offspring per year. They live 30, 40 years sometimes. What they have is very similar to us. They have furin inside of their cells like us humans. So a furin cleavage site that works in us humans also works in a bat. They have an ACE2 receptor, very similar to us. There's a lot of eerie similarities between a bat and a human. So that's where kind of the crossover comes between virologist and bat research. They wanted to use live bats to create vaccines. They wanted to replicate, the human body by essentially studying a bat's body. And if you inject Marburg into a bat, it just kind of winks at you. It smiles. If you inject Marburg into a human, we're dead within hours. So that's why, virologists, that's why there's a fruit bat colony inside the FLI lab in Germany, because they're injecting it with Marburg. They're injecting it with Ebola, and they're watching the immune system of a bat take this, like there's not like it's a walk in the park. They don't even are affected by these viruses that are so lethal to us humans, but we have so much in common. They have livers like us, they have kidneys like us, they have lungs like us, they have hearts like us. And why is their immune system so strong? And this was the obsession of Christian Drosten and all the other virologists of the world in 2015, 16, 17, 18. Why are these bats surviving these lethal human viruses? If you just sit around all day and thinking about viruses, you really are interested in what survives what immune system can survive this virus, and you can learn a lot from a bat's immune system. So this is kind of where the industry is headed towards around the time of a lab leak was trying to create these vaccines, these prototypes, these antigens using bat research, live bat research. So that's kind of where this leads. And, when I want to drive home the point that, it's not a virus, that we dealt with, it was a vaccine. So the same people that created the virus are the same people that created the vaccine. Virologists are glorified veterinarians. You take your dog to a vet, to get for treatment. These people, you take, your live bat to this kind of vet. They're virologists. They are just veterinarians. Is all they are. They study animals. They don't study humans. You can't study humans in a lab. A, it's immoral. B, it's just impossible. No one signs up for these things really, really anymore. But we do have to admit that, these people that did this to us, are kind of laughing at us because they can't, because we can't figure it out. We seem to think that, we're the only species on the planet, but there's 1,400 species of bats, which are, 1,400 times more than we are. That's essentially, I think, where the story of SARS-CoV-2 is. These high-priced, high-flying virologists, studying lab animals, turned us into their lab animal.
We are all bats now.
We are the bats.
Yeah.
You get it. They
Yeah.
turned us into their lab animals, and they're I want to keep driving it home that they're laughing at us because we couldn't figure it out. I think, this is as close to the truth as we can get,
But aren't
if
they
we just-
nervous? You really think they're laughing at us like they will never solve that riddle? But don't you think with all these hearings that are right now happening, that they're nervous?
I think this year they're nervous. This year was a big year for lab leak evidence because it's not in China, anymore. It's in the American files. And in particular, it's what they were doing when this thing leaked. The latest is some of the high, ups in NIAID, the Washington D.C. Bureau for Biomedical Research. They're using Gmail accounts. They went quickly into an underground world of Slack messages and Gmail, and they were nervous at first, and then they realized none of us could figure it out, and they got pretty brazen. They went back on stage. They're back handing out awards to each other right now. And were they laughing at us? Yes. Are they still laughing at us? I think they're laughing a little less right now because a lot's come out.
Yeah, a lot, like weekly new evidence. And yeah, I want to dive a little bit more deeply into your theory. We have talked about the things that most lab leakers would agree upon. And I want to know what is the basis of your theory and what makes it a little bit different from the others. If I could paraphrase it, tell me if I'm wrong. The origin of COVID-19 is not in China, but in the US. The virus leaked from Danielle Anderson's BSL-4 and not Zhengli Shi's BSL-2. And that SARS-CoV-2, you already said that, was a self-transmissible bat vaccine that did not transmit in most types of bats, except Egyptian fruit bats and some other lab animals we talk about later, but it transmits perfectly in humans. It was never designed to kill humans, but to spread very fast. That's why we were saying we are all bats now. Barrick built it, Munster aerosolized it, and it leaked in Wuhan in the BSL-4 under Danielle Anderson. Munster, according to a theory, aerosolized Barrick's genome into an airborne transmissible vaccine. And you say the intermediary species for SARS-CoV-2 will be found in the Rocky Mountain labs. So this is like a short presentation of what your basic theory is. Is it correct?
It's correct. So Christian Drosten said there's a smoking gun for a lab leak, but it was not funded. It was called DARPA DEFUSE. And he said at the time an American university was actually going to insert that fear and cleavage site we were talking about. That's the University of North Carolina and Ralph Barrick. That's why I like Drosten. He's pointing this document like Jim keeps talking about. We keep talking about DARPA DEFUSE is a smoking gun, but it was not funded. That was the defense Christian Drosten used to say there's nothing to see here. Our industry's safe. We didn't blow up the world. And if you look closely, this is a 2018 bid into DARPA, which is the Department of Defense, that the American military, is looking to vaccinate bats from pathogens like SARS so they won't affect operations in Asia. The American empire is strong, for better or for worse, and essentially there's a giant regiment of U.S. troops over there in Asia, and they lost bases. They had to shut down during SARS-1 in 2003. Nipah, Hydra, these can also be weaponized. So there's a dual use aspect to this. Essentially, the enemy, China, North Korea, could weaponize an agent and infect, the U.S. military. And, nothing's stopping the U.S. military. Ain't no tank, ain't no plane, but a pathogen can and will. So there's a big money in American biodefense. And I don't think the Christian Drossens of the world appreciate that. He says it wasn't funded. Everything gets funded in Washington, D.C. To give you some numbers, it was a $4 trillion a year government. Now it's $6. It's the largest government on the planet. I don't want to get too far off into the politics, but I'm just giving you numbers. The NIH, the NIAID, the Anthony Fauci institution is called HHS. It's 1.4 trillion, which is like bigger than the French government, and the type of institution that would fund DARPA DEFUSE. So my opinion is $14 million is a lot of money for most people. It's nothing for the U.S. government. It was just loose couch change. And if you look closely in 2019, the same people that were proposing in 2018 are going forward. They're continuing with their research. The suspects are not stopping. There's big dollars in this. So that's where I diverge from Drosten. He says that's a smoking gun, but it wasn't funded. I say it's a smoking gun because it was funded, because everything gets funded. There's a lot of troops in the Middle East. There's a lot of troops over there in Southeast Asia. There's a lot of troops in Germany still. These virologists, around the world, not just American, just, like the Eddie Holmes in Australia. I'm sure the Christian Drossens of the world, everyone in virology was involved in DARPA type projects. My theory, to make it long and short, was that the DARPA DEFUSE project was funded by Anthony Fauci, the guy that literally, everyone ran to, at least in America, was the one that created this thing, more or less. And it was created in his own personal biodefense lab in Montana, and it was shipped off to Wuhan for testing. Where I focus on the BSL-4 lab. There's two labs in Wuhan. Most people focus on. There's one lab in Wuhan. Most people focus on. It's called a BSL-2. It's like a face mask, maybe like an apron. You go in. You don't have the spacesuit, and you're doing low level research. I think it's just a total waste of time if you want to think this thing came from BSL-2. It floats 60 feet through air. That's 20 meters. It'll infect you in less than 15 minutes. And it doesn't even want you to know it's there. It is very high level engineering that allowed that to happen. And when you're testing something that contagious, it's in a BSL-4. That's the spacesuit. That's the moon suits. You go in, you suit up, they call it. You can't go to the bathroom. There is no bathroom inside of a BSL-4. There's no toilet break. There's no there's no eating because you're in a spacesuit. You are basically an astronaut. It's exclusive territory. Very few people get to go into these BSL-4s. You have to have background check, health check. You got to be in perfect condition. So they work in autonomy, and they work without any oversight, without anyone looking over the shoulder. There's no regulator. There's no FOIA. No one can go inside these labs. It's the best of the best. It's the cream of the crop. And inside this BSL-4 lab in Wuhan was a Westerner, and her name was Danielle Anderson. Bless her heart. But I am 99.999% sure she's our patient zero. This is essentially the other female in Wuhan is the only other one to have lied to us. Lie is a strong word, misled. She told us she was working on Ebola when she was in that BSL-4. I've looked at her long resume. It's about 100 papers. She's the cream of the crop. It's about 100 papers. There's zero papers on Ebola. She actually is doing a lot of coronavirus research towards the end of 2019, and she's cross-referencing with some of the people in the DARPA DEFUSE document in Montana and North Carolina. So she's doing some work with them, for them. It's so precise, we can actually see her movements around the world in 2018 and 2019 because we have her cell phone. Her cell phone was part of the intel that came out. American intel was spying, and it released the phone messages. So there's a cell phone report that shows a lack of activity in October 2019 around the BSL-4 and just the BSL-4. It's like every cell phone just scattered. There's nothing there. October 1st, it's a beehive of operation. On October 10th, there's no one. There's no one near that BSL-4 lab. And that's what really, I like the timeline that it shows, and I like the phone that it shows. And of all those phones, there's not a lot, but there's about a dozen phones. One of those dozen phones was on the DARPA DEFUSE proposal. That to me is a very, important piece of this puzzle, that you have a phone that was on the DARPA DEFUSE proposal inside the Wuhan BSL-4, and it vacated. It vacated the premise in October 10th. And it's hard to see where she's at. I think she's self-isolating in her downtown dormitory. This tiny little dorm room that Danielle Anderson's self-isolating in is the epicenter of an outbreak. And we know that because of Weibo. It's kind of the Chinese Twitter. And on there, some scientists went in there and traced back the earliest outbreak. It was like early December, but it was based on Weibo. It's this tiny little dorm room inside downtown Wuhan. And that dorm room in downtown Wuhan is the same dorm room she's living at. This woman that was on DARPA DEFUSE, who misled us and told us she was working on Ebola, as basically, her story's not adding up. Whereas opposed to her story, the other one is, Shi Zhengli, the Chinese bat lady. Her story, it's complex, but I've checked into it. I've wasted way too much time on this thing. She had bad paperwork. She's guilty of that. She had sloppy paperwork, but almost everything she said checked out, and she fights. She's a fighter. She gets in front of the cameras and she says, I did not do this. I did not come from my lab. It's just like, if she's lying, she's one of the world's best. But this other woman, it's not her fault because it leaked. She didn't create it, by the way. She's the first victim of us all. If it leaked, it was on her watch at least. And she's adamant she was not sick. She's a 40-year-old runner. She uses this thing called Strava. Strava is a runner's app on your cell phone. When you track your miles when you're running, You're broadcasting to the working world where you are. The only reason we know where she was is because of her Strava app, probably, along with a few other apps. On your phone in the background, it's pinging a cell phone tower showing you I'm here. And it showed that she was in Wuhan in the BSL-4 from June of 2019 until October. She leaves in October, but then her and her boss, Linfa Wang, come back in November. I have a picture of her inside the Wuhan BSL-4. It's almost like a hostage picture. The way she's just kind of posing in front of the security camera. She's got one of her colleagues. I'm not sure who it is, but it's her in one, and it's someone else in another spacesuit. And she's just looking at the security camera, and I'm guessing it's her boss, Linfa Wang, taking a picture of it. And it's almost as like it's a hostage video. It's a timestamp. It says November 29th, 2019. That's her last day in the Wuhan BSL-4, November 2019. And she never explained why she left Wuhan after 4 years of living there off and on. She's a part-time employee at the Wuhan Institute of Virology. She's the only Westerner in all of Wuhan at the Wuhan Institute of Virology, as best I can tell. A lot of other, Christian Trossen's been there, Ralph Barrett, Peter Daszak, they all fly in, but they fly in, they fly out, they visit. She's living there because she's researching there. She's researching live bats inside that Wuhan BSL-4. And her boss is Chinese, but they live in Singapore, and his name's Linfa Wang. And I guess to jump forward a little bit, on January 10th, 2020, Linfa Wang stepped down from his position as the director of Duke's Emerging Infectious Disease Program, as if to say he was inside the Wuhan Institute of Virology. I vouch for her. It never made sense to me why she's in there. Her story never really added up. Her story came out in June of 2021, before DARPA DEFUSE leaked. So just to go back, there's a big debate about at least in the US, about lab leak in May of 2021. Then June, we got the Fauci emails. We got the Christian G. Anderson emailing Anthony Fauci. We find the genome inconsistent with expectations of evolutionary theory. It's engineered. The same guys that said that wrote it's the not engineered paper called Approximate Origins. And we're all like spinning, what's going on here? Anthony Fauci's involved in this. This thing goes all the way to the top. He's the president of biodefense in America. He is the czar of America's, emerging infectious disease program. He has a $6 billion budget, twice the size of DARPA. It's a huge one. It's bigger. It's the best. It's got all the money in the world just for biodefense, $6 billion. So going back to what Christian Drosten said, everything gets funded. My theory is in 2019. It's not my theory, it's their words. We're going to resubmit this DARPA DEFUSE proposal under not the Department of Defense. We're going to move it into the Anthony Fauci, HHS, the NIH, the NIAID, it's called, this giant alphabet soup of, healthcare. Not like you and I healthcare. This virus has nothing to do with us. It's animal healthcare. It's biodefense. It's studying these emerging infectious diseases from bats. And that's what Danielle Anderson was doing. She was an American contractor. She's Australian passport, living in Singapore, temporarily stationed in Wuhan, bidding on a giant for $82 million Anthony Fauci project in 2019. So her dates, her movements correlate perfectly with the bid for the NIH version of DARPA DEFUSE. So she is our patient zero if you're willing to listen to the evidence.
And how can she be infected when she's working in the BSL-4 and the experiment kind of failed? This is also part of your theory that she went there, she tried to infect the bats and wanted to see if they transmit, and they didn't transmit. But how do you think this leak towards her could have happened?
I don't know. I don't think she knows. She's telling you that she's adamant she was not sick. It's almost as if to say, I didn't do it. It could have been her assistant. It could have been her friend. It could have been, she doesn't even know. She doesn't know how it got out. But inside this BSL-4 is her experiment, which turned into SARS-CoV-2. Once you get your brain around everything, it's so overwhelming. It's almost funny. Essentially, what she's playing around with inside that BSL-4 lab is something that's been developed in an American lab. And I'll get to that. We'll get to that later. But she's in there trying to take this American, we'll call it formula, serum, vaccine, and she's trying to jab it into these tiny little Chinese bats. And when you're jabbing it into a Chinese little bat, it's a long needle. It's very easy to stick yourself with a needle. I don't care if you have a spacesuit on, 2 gloves on, metal spacesuit on, you can still puncture yourself. It's very common for a needle stick to occur. That's the term, needle stick. You put the needle into your own body. It's supposed to go into the bat. It's supposed to go into the lab animal, but it winds up sticking yourself. And if you stick yourself with that needle, bad things happen. Either you die or someone else dies. So they have what's called like a 2-person rule, but she's on a headset. She's a loner. She is a loner inside Wuhan. As far as I can tell, she has no colleagues, no friends. She's kind of trying to, cover her tracks. She made it sound as if she's alone in there. She's on a headset talking to a technician in another room. She is the cream of the crop. If there's only about 5 people, 6 people qualified to go into that BSL-4. Shi Zhengli, she wasn't in there. Daniel Anderson, and then that leaves about 4 others that could go in there with her. I believe strongly she's in there alone. She's a workaholic. She's probably working 7 days a week. And the way she describes it in the news article, she's alone. She has a headpiece. So the lab is so new. It was built by the French. Once you read about it, which I did too much, you quickly realize it's not used very often. It was used by her mainly. She's alone in there, bless her heart, and she was given the impossible task to test a contagious vaccine inside of a foreign lab on foreign soil. And she had to go there. They had to go there because that's the only place where these bats could be kept alive in captivity, was in Wuhan. These bats are very special. Again, Christian Drosten will tell you why they're special, because it's the source of SARS-1. The biodefense industry will tell you they're special. They were obsessed with these little tiny bats. That little pathogen called SARS was pretty lethal, a lot more so than SARS-2. It could kill you. It was so lethal, it burned out. That's the thing with SARS-2. It's just not lethal. But this thing was designed to spread. If you think about it, why on earth would someone design something to spread? Until you read a document, then it makes sense, because you're trying to vaccinate an animal, a remote animal reservoir, an animal that doesn't hang out with humans. It hangs out in caves in the deep jungles of Cambodia and Asia and Laos and all these other places. And you can't go around and inject each bat each time you're doing it. It's impossible. It's the latest and greatest thing, according to a Max Planck German scientist I learned from, Guy Reeves, was called self-spreading vaccines, contagious vaccines, transmissible vaccines. These are viruses designed to spread, but they're not viruses, they're vaccines. They have high transmissibility and low virulence. Doesn't that sound familiar? I mean, for the past 4 years, we've been having to deal with something with high transmissibility but low virulence. For the no virus crowd out there, I agree with you. It's not a virus. It was the vaccine. You took the vaccine, whether you wanted it or not. An automated, transmissible airborne vaccine, an agent designed to inoculate, not infect. You got an inoculation. And then some people come back and say, well, I got infected twice. Well, that's even part of the feature of this thing. It's designed to reinfect. It's part of the immoral side of what virology was doing in 2016, 17, 18, 19. And I'll admit it's very, very hard to even comprehend that.